So this is so today we'll be having the week 9 of the uh, course of the session for the course Introduction to Proteogenomics. So till now what we have looked at the individual level, how epigenomics, genomics technologies can be taken way forward, can be analyzed, can be sequenced, and then can be taken forward. So now what we are doing is that we will be, and also we have looked at the proteomics technologies, specifically the mass spectrometry based proteomics technologies, that how it became quite powerful and how it can help us in analyzing the protein sequences, also help us in the quantification procedure, or how it helps us in the quantification for the proteomes or the all the proteins present in a particular human body or a cell or a tissue or an organ, and also the how the after the human genome maps, how your proteome maps have taken place through antibody based approaches, through your mass spectrometry based approaches. But now today we will be looking at the most interesting and the most part of which we are studying the course is the proteogenomics. So we are saying from the first week that we should appreciate that how those two techniques, amalgamation of those two techniques, help us to understand the pathophysiology better, pathogen better, the disease better. So today we will look at certain aspects of it, how it can be done, what are the ways that can be integrated, and how both the genomic sequencing data, next generation sequencing data, and the proteomics data can be cumulatively can be used together for better understanding. So now if we look at the concepts prevailing integration of the genomics and the proteomics data, also we will at the end of today's session we will try to brush up a little bit about the proteomics technologies, the proteomics quantification technologies using the labeling technologies. So just brush up a little bit, because ultimately we need to identify the peptide sequences or the protein sequences through mass spectrometry, and how we can quantify in a better way, or how we can increase the coverage, because the genomic sequences will give us the information about all the genes, all the codons, all the exomes, all the exomes. But how we can increase the coverage of the protein sequences is still a challenge, and on which the whole community is looking forward, the ways forward to how to accurately quantify, increase the coverage and accurately quantify the protein or the and identify the peptide sequences. So we will discuss some case studies and those kind of studies as well. So how specifically, so if you are given an idea, that if you are asked that how you can integrate the genomics and the proteomics data. So first of all, so for example, if you are taking a patient, so now is the concept of the so I hope that you all are well aware, that well versed with the techniques like immunotherapy, personalized medicine, precision medicine. But how you are going to do that, or how it can become a reality. So for example, now if you it is a patient for any cancer, for example, you take, and you want to have develop or administer an immunotherapy or therapy for this, design of administer a vaccine, design a vaccine for this patient. So how you are going to do that? So firstly you will be taking up the tumor tissues, then you can perform the whole exome and the transcriptome sequencing. So now till now what we were discussing, so if you remember that in the first week, or the first or the second week, we had discussed about the workflows at the CPTAC, that how your whole exome and the whole proteome sequencing can be uh, uh, together, used together for the have been and is being very much being aggressively helping us to uh, understand the relevance of the data. So now we will be looking at the parts, the bits and process, that the uh, nitty gritty of those things, that how we can integrate those. So you have your whole exome and the transcriptome sequencing. So what you will get, so if I ask you that what you will get from whole exome and the transcriptome sequencing. So whole exome, you will say that you will firstly that why the difference, you should be cleared about the difference between your whole genome sequencing and the whole exome sequencing. So in the whole genome sequencing, you will be capturing the whole end-to-end genomes for an individual, but in the whole exome you are only capturing the exon part, exonic region of the eukaryotic DNA sequence. So fully so now the problem and the pros and cons of both the things are that in the whole genome sequencing you would be able to capture both the germline and the somatic variations. But when you are doing the whole exome sequencing, so you are capturing only the somatic variations and not the mutation. So you need to be clear that what because of the with the knowing that what with what objective you are going to do, or you are trying to analyze, or what you are trying to achieve will depend upon the course of the sequencing you will be performing and the sequencing analysis you will be doing. So now so we have discussed that in the whole exome sequencing you have got your sequences, then you will be so I'll, if you all don't recall, just go back to the previous videos or the and try to recall it that how we can we will be firstly aligning with the human genome. So now you have the patient and you want to do it in a personalized way, then what will be your approach? So the one way it is always an universal approach is that you take the tumor tissue, you take the whole exome or the whole genome, and then try to match with the HG38 or HG19 or HG18, whatever suits to you. So HG38 is the more recent version of the normal human genome what is present, but that will not give you exact, because that will not give you the patient which the patient mutations, because you know that the people generally carries a lot of mutations, but they are not deleterious or the harmful for that particular person. There are many reasons for that, because the our body's immune cells tries to recognize all those proteins which are not of normal size, normal shape, or normal functionalities, and try to eradicate those. So, our body's immune system, a personal immune system, uh, is very much well versed with how your protein sequences work, or what are the normal functional protein sequences. Now if we match with a sequence for an HG38 or HG19 and found out some mutation, but that mutation may not be harmful, and designing a drug or a vaccine against that mutation might not be so much useful. So for that what we need is that we need to have a whole exome sequence from the tumor tissues, as well as the normal from the peritumoral region or the PBMCs, that is your peripheral blood mononuclear cells, which contains the normal DNA. Okay, so now what you will be doing is that you will be trying to aligning with that. So then what will happen is that you will get an exact what are the differences and the mutations that are present in that particular person. So now we will detect the somatic mutation. So since we are doing whole exome sequencing, so that's why you will be getting the uh, uh, whole exome sequencing. So now this is one thing. So now another thing is that you will take the proteins. So here, since we are looking for so this article aimed for understanding of the some immunogenic candidates or identification of the immunogenic candidates. So that's why it is written that you will be immunoprecipitating using HLA class 1 and class 2 complex. Otherwise, with the simple workflow of the proteomics that we generally follow is that splice the cells or the tissues, and then what we do is that we extract our proteins, and after extracting out the proteins we give it. So I hope you all are well acquainted with the terms like bottom up proteomics by now, where we break down our proteins into digest it into peptides via proteolytic enzymes like your trypsin or other enzymes, and then after digestion we inject that into the mass spectrometer and detects the imbases of those particular peptides, which then through which we identify our proteins. So now there is a now part that if I say these are so this for anyone, if we tell these two things, so it will be like your two stories for a person, two different stories. That one in one case, in one tube, one is extracting out the DNA, extracting out the RNA, doing the whole exome transcriptome, doing the matching the variants, finding the variants, deporting the mutations. Another story can be extracting on the proteins, doing the tumor versus what is the different proteome expression profile. And these were the two different stories with people of both the groups being very much happy and getting the results. But you have to understand that it is so the scientific community could understand that it is not getting the exact result what we are looking for. Now if we need to integrate both the data or both the parts of the story into one, so there you need to have a common factor that where you will be linking that. Now in this whole two process, so the sequencing technology and the proteomics technologies are quite independent on their own. But if you understand their usage properly, then you will be understanding that there is a common link, and that common link is a database. So how the database is linked to it? So for instance, if you uh, whenever do a proteomic search, so we have discussed quite detail like the mascot, if you remember, that where we are giving a contaminants database, a swiss prot database, or an organism specific database. We are uploading the raw file, so the mass spec will acquire its data irrespective of any database. So it will just calculate your embases. Now your embases belong to a peptide, you belong to some molecule, belong to a certain thing. So that mass spec has nothing to do with it. Similarly, sequencing technologies will generate your ATGC sequence, read your sequence, and will give you the data, and it has nothing to do with it that whether it is your variant or whether it is anything. So it all doesn't depend on all these things. But your mass spec data, once the raw files are generated, so now you have got the raw files. So now how we will be analyzing that, so that becomes quite challenging and important. So if you remember from the mascot, so we need to have provide a database. Similarly, for any search engine you go, maxquant, andromeda, PD, fracpipe, any you take, any software you take for that matter, irrespective of the software, you need to have a database. So why you need to have a database? Because the embases that you have got, you need to match with certain things to identify that which ions, then which peptides, and then which proteins. So how will you get that? So we generally, we generally proteomics workflow, during the mass spec based proteomics workflow, what it has happened is that you will give the organism specific database either from Uniprot, either from Swissprot. You will provide that. So after you provide that database, so what will happen that the software has inbuilt in silico way, it will uh, get the protein sequences, get the MYZ spectra, and then try to match with whatever you have acquired through the data, and adjusting your certain mass tolerances and certain parameters and all those. So it will give you the with certain confidence, certain level of confidence, that this can be a particular peptide, this can be a particular protein. Then you need to mention your threshold criteria, and you then can finally go ahead with whatever confident peptides and the proteins you have identified. So now this was the best chance where one can integrate this thing. So once you have those somatic mutation detected, so now you have a person's whole genome sequence data, whole RNA sequence data, and you have aligned it, you have got the mutation specific. So now you can generate your own database which will contain your all mutations. So all those somatic mutations that one was looking only at the level of the genomic level, at the variants level. Now, whether it has been translated at the protein level or not, whether if at all it has been translated at the proteome level, what is its effect, or whether that proteins, because of the missense variations or the nonsense variations or the truncated protein sequence, because of that, if its functionality has been changed, can it be used for an immunotherapeutic treatment? So now you know. So, and if you develop a treatment against that particular protein, so that would be 100% safe. First of all, there would be no adverse reaction, because the body will identify only that protein present in that tumor region, which is showing that uh, effect, or the effect of that, because of that particular mutation. So that will lead you to the, for your better, first of all, better treatment opportunities, therapeutic opportunities, better modalities, also the effect, that whether it has been really translated or not. So in the central dogma, you know that from the DNA, then the transcription to the RNA, then to the translation to the protein, and then to the modifications. So how it has been, whether it has been affected, whether it has been changed in this process, so all this can be easily known through this. So this is where the customized database construction procedure becomes very much crucial and very much necessary for us to understand, and helps us in integration. uh, so this whole integration of the genomics and proteomics depends on the customized database construction. So this step helps us in integrating the both the technologies. And then finally, identification of a tumor specific. You can identify mutant peptides. You can identify any such peptides. So how, so that's the thing what I was explaining. So the msms data, so this data, this raw files does know nothing if you don't perform an analysis, if you don't provide it a reference spectra, don't provide it a reference database, it won't be able to match with your known database and help you to gain or identify those peptides. So now, so the database plus the search engine. So as I was mentioning, mascot, sequest, you can have any of the search engines, any of the popular search engines that are being used daily. So the database can be, now you have, if you have your, you can make your own database, as well as there are some database of the single nucleotide polymorphisms that are generally being uh, present, that are being present as a resource. You can use that. For example, DBSNP variant peptides, cancer related variant peptides you can provide. So we will be looking into it also, that how the different peptides and the proteins which are reported in the literature. But again saying that we are looking, now thinking much beyond. So we are looking at much beyond of our uh, imagination, of not only looking at the universal things but at the personal level, personalized medicine, personalized therapeutics, personalized treatments, and to assess at the personal level, that whether how a person will be responding to a particular therapy, how a person will be uh, and how a novel treatment can be developed for a particular uh, person. So now you can see that you the search results. So confidence evaluation of your wild type peptides. So as I was mentioning, so the proteomics software will give you. So uh, once it is matching your proteomics data. So now what it will be doing is that it will be trying to it provides peptides and the proteins with certain uh, uh, with your certain confidence, that how much it is matching, and with the false discovery rates and the p values and all this. So similarly, you will be having confidence evaluation of the variant peptides, and then you will have the identified variant peptides. Then also the thing is that you should go for uh, variant peptides. What you say is that uh, try to validate those. So how we will be validating those? So we have already discussed about the targeted proteomics based approaches, SRM, PRM. So I hope you all are acquainted with it. If you're not, then just please refresh it once. So you will be getting that how you can identify. So we'll be discussing a case study as well. So yeah, so as I was mentioning the case study, so this is a paper. So till now, what we have done is that we were discussing much about the cancer, human tissues, human things. So before going into the human things, so human is the cancer and all is quite needed, important for us to understand, but also whether this approach. So I think that if you all are working with any microbial diseases or any microbiological or virus related things, and obviously after the COVID. So you all might be thinking that whether this approach can be used for understanding microbial diseases or not. So this is a case study. This is a paper from our lab where we have tried to identify the. So you can see that the detection of the mutant peptides. So I think it needs no introduction to you all for the COVID and the mutations. So what we know till now, what the whole world focused on was the mutations at the genomic level, that the strain is getting changed, and the how this thing is getting changed. So you will be having an RT-PCR, and if you find out, so sometimes it gets skipped also, so resulting in your false negative and all, because the if it is a lot of change in the strain, so it won't be getting amplified, and you will have a less uh, you will have a high city value and all those things. So people were just trying to trace it out, and the variance, I think that till now it is prevalent, might be in some parts of the world where you are getting certain variants in the genomic level. So now what we are, we all know that the variants at the genomic level, but that this genome gets viral genomes gets integrated, and the detection of those particular peptides. So now if we can detect the mutant at the peptide level of those SARS-CoV-2 variants by LCMS in the DDA approach using an in-house database. So what concept that we were talking about in the of the construction of the customized database for the understanding of the human proteins for the tumor specific diseases. So now here we showed that we can detect some mutant peptides of the SARS-CoV-2 variants in using an in-house database, and also that too from a clinical samples. So clinical samples means from a person affected with COVID positive, from his or her swab or plasma, we can detect the mutant peptide. So now from there you can understand that you can correlate. So if this thing is approached, and this thing is uh, taken in routine use, then you can from the directly from the patient, you can try to identify that for which peptide or due to which protein level which peptides are causing a person to become severe or a non-severe. So and also the effects of this particular protein. So I'll just say you tell you something that, so since though the world monitors the effects of the variants by the genomic level, but all those mutations, you also will agree with to that fact that all not are deleterious, and some are quite very much dangerous, and they become quite dangerous. So the reason is that all the effects that happen are at the protein level. So the virus that ultimately encompasses, or ultimately that structures that bypasses our immune system, because our immune system can recognize certain proteins and can act upon it, but whatever fights against our immune system or bypasses it are the structural and the protein level. And also you might have, I'm not sure that if you all are aware that whatever uh, more deaths that were occurring during the COVID time were due to the enhanced immune system or your cytokines and all those things, which was causing harm to the one person's own lung cells if you cannot control the cytokine levels and the all those things. So that also depends on the proteins against which the immune system is fighting. So it becomes quite important and crucial aspect for us to try to understand that what is happening at the peptide level. So if you all are interested, you can go and read this article, but here I will show that how we have done. So the concept of the mutant peptides that are being taken, obviously we will discuss in great detail uh, about how it is happening in the cancer database. So now how simply with the simple concepts have tried to identify the peptides from the clinical samples of the SARS-CoV-2. So where we'll be discussing. So here, so as you know that there was a huge rush, means the genomic sequences of SARS-CoV-2 across the world, across the globe, across the labs, what daily was being reposited, and with all the variants databases. So the SARS-CoV-2 database that contains uh, not only from entire world but from India also, it contained a huge number. So what we do, so GSAID was one of the initiative that was taken for sharing of the if you say that the sharing of the resources or the genomic sequences for the entire influenza or the infectious diseases, but it became in very much highlighted during the COVID time because all the COVID sequences were getting deposited in the GSAID. So GSAID is a global initiative, so you can now also go and search. So there you will be finding so all those SARS-CoV-2 sequences. So what we did, so our patients were from obviously from India and specifically from Mumbai. So SARS-CoV-2 patient isolated genomic sequences. So whatever sequences were present for the country India, so we downloaded that. So now how? So now I'll say you. So just think of the scenario where we have the patient, where we have the collected the clinical specimens, the fluids and the plasma. So their proteins have been extracted. So that protein contains your both host proteins, that is your human proteins, plasma and the swab proteins, as well as the proteins from the viruses. But there you need to understand there is a challenge of identifying. So we are here trying to identify not the host but the virus peptides. So the virus proteins will be quite less in number in comparison to the host proteins. So the coverage will be quite low, so it will get masked in the mass spectrometer. So it is quite less chance to get digested and to identify. So the better approach is to enrich those proteins. So how will you enrich those? So you will have an antibody. So I am just giving you the experimental conditions and the experimental thought processes as well that will that leads us to the better understanding of it. So you will need some antibodies against your nucleoprotein, against your replicase proteins, and then you will try to pull those down. So that would yield more low abundant proteins. And if you fractionate those, so the an ideal condition would be if you fractionate those and then run in the mass spectrometer each fractions after you get all those proteins, then what you will find out is that uh, so you will get a better coverage. But again, you have to understand that more process you will add, so more it will be a cost. The cost will be associated with it. The more time it will take, the more labor it will take. And in the situations like a pandemic, you can understand that you need more robust, more fast, more reproducible, but in a very fast and less time consuming and less labor. So you can understand. So you cannot always go for ideal experimental conditions, and you need to have a trade-off between ideal and what is your most practical and experimental requirements. Uh, so you need to understand that. So what we can do is that so you need to have those setup in mind. So always you can think of that we cannot identify everything in if you want to do quick, but at least we need to identify and we try to find out that whether we are able to do or not. So we had downloaded all those databases. So now what we will do is that, so now so as I was telling about the situation, so we have the proteins peptides run in the mass spec. So as I was the chromatogram with the MYZ files, now that contains of the host protein, virus proteins, mutated protein, non-mutated proteins, we have no idea. So now we generally provide a database. So now if you provide a database of the human sequence, so it will match with the theoretical human spectra with the mass spectra that is getting. And then what it will do is that it will give you the proteins. Now if you provide with a virus sequence database that was present at that time in the uniprot, so it will give the viral peptides, but we want the mutated peptides. So how we will get the mutation peptides, because we cannot do it in our own. So if you think, so any random answer or a best answer would be to randomly insert the mutations, or to get the mutations list and try to incorporate individually, but that was not possible, or that is not feasible. So we had taken that sequences, genomic sequences, from their in silico translation we did in the 3 frames. So you can see that if the 3 frames, if we perform the translation, so we will get 3 frames of the proteins. But that would again we will have a quite a big number, means the size of the database would become huge, and again the searching and the database searching would take time. Also the chances of false identification would increase, so you have to understand. So the more number of protein sequences we give, more number of theoretical spectra which might get identified. So always think that the false discovery is like the that it is identified by chance. So we don't need any identification or any claims by chance. We need our identifications to be foolproof, our identifications to be confident that it is not by chance, but it is there, that's why it is getting. So after we got the protein sequences, now what we did is that we matched with the original spike sequences. So for example, so if it is for a spike proteins particular sequence, we have got from all the so around if I could remember, around uh, 25,000 or something, I can't remember the number of sequences was there for each sequence, for each region, spikes region, we are getting into 3 number of sequences, protein sequences. So we cannot, we can put this directly into the search engine, but that would have increased our uh, uh, false identification levels. So what we are doing is that we are trying to reducing it. So why, so how we are doing that? So we are giving the less number of sequences. So for that, what we have done, what strategy we have taken? The strategy we taken as that we had taken the original spike sequence, that is the Wuhan strain, which did not have any mutations, and then matched with whatever we are getting. So the virus can be generally in the eukaryotic scenario, we take the 6 frame translation uh, because of the how the translation happens. But in this case, uh, 3 frame, within 3 frame, uh, we get the protein sequence that is being translated. So now once we got the 3 frame translated one protein sequence, so now which frame is the correct one? So second question is that which frame is the correct one? So for that one strain's original sequence, for example, spike sequence, we take that and we align that. So once we align that, so our mutation or the thing that we were thinking of or we were looking of was one single amino acid change. So the majority, the whichever is the right translation, right frame translation, so it will be getting matched. And once it gets matched means match means having the highest number of alignment with certain mutations, it might be 100% match that there was no mutation, or it might be 99%, 98%. So we chose only those sequences that was giving the highest match with the original strain, because the mutations are at specific level and it is not 100% mutated. So in this way we reduced our false discovery chances and the databases became less. After that we searched using our database. So here it's all written, and if the article, if you go through it, and also the codes which were all written by us. So all these things are available, so you can look and check to it. So then what we did is that now the database is fine. So now we searched it in the using the uh, database for the with the mass spectrometry data, and then it got identified some of your peptides which are mutated. For example, here, so I'll just explain it and I'll also show. So you can see that this is the original peptide from a nucleoprotein. Original means the Wuhan strain one, which is sort of the first one reported to be reported, and it was not mutated. The what is known to everyone that it is not mutated, though actually we don't know, but still what is considered as to be the first reference genome sequence of the Wuhan strain 1. And here you can see that in respective of H, there was a Q that was detected by our search engine because of the from the databases. So now see that we could identify some peptides, but now there's a question that how will you validate that, because as I was mentioning that the proteomics data first itself contains a huge uh, noises. Secondly, you are doing it from a uh, human host clinical sample, then you are identifying a peptide. Though we could reduce the number of database, but still we are translating it in silico way. So the identification of that particular peptide becomes quite the claim of having identifying such a peptide becomes quite challenging, and it doesn't become quite acceptable without validating it. So that's why from global discovery to targeted proteomics is quite necessary to understand, and also to validate our claims of the identification of the mutated peptides, specifically in this kind of clinical applications. So here what we could show them, that you can see that the if you understand the concept of MRM, so MRM is a very robust and controlled way of validating the peptides. Why? So if you don't I'm just brushing up the concept once again. Else you can revisit that particular week's video and go through it. So here what you do is that you give a peptide that is a precursor, and you calculate theoretically your transitions, that is your precursor will be fragmented on a particular collision energy, and that precursor product ion, that is the transition. So you calculate it and give the instrument that for my peptide these are my transitions. So can you detect it? So you ask the instrument that are you able to detect this particular Q1 Q3 pair or the precursor product ion pair? So it will give you the peak once it the transition gets that particular pair, which is for that particular peptide. So the M by Z for this particular peptide, if the amino acid was H, would be 1, would be something, and if it is Q, then it would be something else. But what is happening, so you can understand that there will be obviously the masses of the two monoisotopic mass of the two amino acids are different, so that's why M by Z would be different. So here there is no doubt, right? So here no one can deny the claim that okay, H and Q is quite different. H and Q is different, so their masses are different, their membranes will be different. Now their products will also be different. So if you feed to the instrument, or if you ask the skyline file for that this particular peptide, then it will give certain precursor product transitions. But if you give by changing H to Q, it will give you certain precursor peptide transition. So now if your instrument can pick that, then you no one can deny your fact that that particular, if this along with this transition it is getting picked up in the mRNA based study, then you can no one can deny the fact that the peptide is that one only. So that why we have done MRM based studies. So to find, so using the wild type peptide and your mutated one peptide, and you can see that in the library also it is getting detected, that in the theoretical spectra library also it is containing the mutated peptide. So it, in this way we could validate certain peptides, showing that it was detected, and we could detect the mutant peptides on the basis of this workflow with the help of the database and the in silico based translations. The simple concept, but can be useful for a clinical level relevant study. Similarly, for some of the proteins for the replicase, and you can in the different samples and the normals and these things different intensities across, so that could be correlated clinically for that whether the severe patients have these mutations or not, or these things. So this is how we, with the help of a simple uh, concept, we could try to detect and try to solve an important problem of those mutated peptides. And this not only restricted to COVID, it can be used extrapolated to any such infectious disease, as well as it can be much more polished in a way that for future any epidemics and pandemics can be taken forward easily. So this is what I first thought of by starting, that only it is that the it's not always key, it's only dependent on humans or cancers or cancer research or tumors only. We can apply the concepts of proteogenomics. We can concept the proteogenomics aspects, though the this though we are focusing much more on the cancer related aspects and all in the proteogenomics approach, but the microbial and the aspects and the uh, specifically the infectious diseases can also be combated or tackled with the proteogenomics approach. So that's why firstly I thought of sharing uh, that simple concepts that we have used and that can be taken forward, and how we could detect certain mutant peptides in COVID, which can be forward taken forward further. So then we will be coming into the for the human or the cancer, that the sequence specific database. So the major thing that depends on, or the integration of these two things is the, that part is the database part. So here the both the technologies can be useful in a way that one can give you the sequence and one depends on a database, because without a database, so mass spec, if it's very much, it contains nothing, or it knows nothing, or you can predict, or you can interpret it out nothing from that. So there comes the concept of the database. So preparing such a database is very much useful, and that's why I explained you all the things that while in working on a real problem or a real application, what things we need to keep in mind. Like the first important concept is to understand that how the algorithm works. Then only you can understand the why. Then, because one can debate that when there is a chance, very less chance of identifying viral peptides or low abundant peptides in that matter, then why not increase our database, or pump our, means give the database a huge amount of precursors, huge amount of theoretical spectra, so that one can get easily matched it up. But we need to understand that we don't want anything junk to get matched it up. We want something concrete which can be validated, as I showed, that which can be validated in your mRNA, PRM, or antibody based approaches like Western blot ELISA, or is actually the particular biomarker, because these studies are quite resource driven, quite time consuming. So we cannot have the privilege to waste our resources to identify something which is not relevant and which could not be validated in later stages. So that's why we need to be extremely cautious. So that's why understanding how the algorithms work is quite important, both the sequencing and both the mass spec identification of the protein. Then only one can build up those customized robust databases which will have less chances of uh, false identification. Also the parameters are quite important, like to keep a FDR of 1%, to check at the protein level, at the peptide level, at the PSMs, how much unique peptides, how much confident Q value, and all those things. So all those things are quite important whenever we design any proteomics experiment or a proteogenomics experiment. So and also these things, the proteomics data sets and all those transcriptomics and all remains quite useful in understanding and predicting. So we need to have understand the data quality, which we have uh, I guess discussed, if not in detail, still in roughly the different your. So you need to have a run to run check, run to run variation using QC pools. You need to check a measure of using different clustering algorithms like PCA, ICA, etc. So to have a understanding, so correlation analysis and all, the whether there is any run to run variations, because these processes are quite cumbersome and goes through a lot of processes, as you all can understand. So now if you want to create a sequence specific database for human, so this is one of the tool that you can try to use. So we had for the COVID thing, we had done just in silico translation, and have just filtered it out on the basis of the known reference sequences. But if from a sequence variant sequence, if you want to have create a database for the proteogenomics, for the applications like the one which I showed here, so you can use a sequence database, that is a custom prod bridges. So the concept is same. So the concept here is also same. So from the VCF file, it does a 6' translation, nothing else. So it provides you a database by just translating it from the variant file. From the variance file, it will annotate your genomic sequence. So so the prokaryotic translations are quite in a way straightforward, But so human sequences, you need to keep in mind your concepts of your specifically your molecular biological concepts of the translations. And here they are doing by the VCF files by annotating the variants. So we will be discussing in the VCF files, that explore the VCF file, what is a how to read your VCF file, because VCF file is quite important. So now again, if you think of the genomic sequence, if you cannot recall, please go for the previous week's videos. And so now you can understand that you have the genomic sequence, you have aligned it. So if you are doing a personalized alignment, what you have done is that you have aligned it using your own human sequence, that is your same PBMCs or other non-tumorous regions or the non-tumor DNA part. So what you are doing is that you are aligning the sequence. Now after alignment, you are getting the uh, variance. Now you will annotate the variance. So now this annotating, after annotating the variance, you are getting a VCF file. So this VCF file is first of all important to understand, to assess that what are the mutations present in your that person, the somatic mutations or the germline mutations or the somatic mutations, that you can target and you can understand. Next, what you know is that you are trying to get it uh, for the database generation. So if you are want to planning to have a proteogenomic study, so you need to have a customized database that will be using up this your VCF file. So reading and understanding the VCF file becomes quite important. So that's what we will be discussing today. So this is the publication, so you can go through it. So this is from the Bing Zhang's from the Broad Institutes, which generate your customized protein database from RNA sequence data for your proteomic search. So what it requires, so first of all, whenever we use any tool or any software, what we try to understand that we need to have an input file, we need to have a output file. So input and output, so what is the file text requirements for the input that we provide, and then what we get as an output data. So here you can see that the custom ProDB requires your binary sequence alignment format. So BAM cannot be visualized. If you want to visualize your alignment, then you need to convert it into the sequence alignment map or the SAM files or the BED format, that is your browser extensible data format. Then only you can visualize. And where you can visualize, so there is a very good popular tool is the integratics genome visualizer, which we will again explore today. So to understand that how you can uh, how you can visualize your chromosomes and your splice junctions and all those in that thing. By so we cannot load any unfortunately we cannot load any bait or the VCF file, but we will be looking or exploring in a way, in a web app also you can download that and try to find out. So the BAM file format, which is in a binary sequence alignment format, so we cannot go and visualize that. So you need to convert it into SAM format or a bed format, and a VCF file as input for each sample of interest using single so GetK, SAM tools, and the genome analysis toolkits. So transcript expression estimates when available. So now expression filter. So for a so it was meant to be for the RNA sequence data, but for fortunately that you can use it for the whole exome or the whole genome sequence data as well. From the VCF file, you can generate the database, customized database, and I have personally used that and to generate the database. So for a given WAF file, you calculate the RPKM function. So that's why through each week we have defined you the concepts, and so that in understanding the integration of these two technologies, it becomes quite easy for you also. The RPKM, again, if you don't recall, go back. Though we have discussed in detail regarding the reads per kilobase, at reads per kilobase per million sequence reads, and then the out process function outputs a FASTA file for proteins with an RPKM value greater than your user defined cutoff. So you will be providing the cutoffs that will be required. So function, again, out shared pro, so these are all functions that they are using. So the input function objects from a VCF file, so it will contains you the informations or the variation related information from one or multiple samples. So this is important, one sample or multiple samples. So if you are going to generate your database, so you can have a person or a people, a person, a diseased person. So you have the normal DNA, diseased part DNA from the diseased part protein, normal protein. Did the VCF file, PRN sequencing VCF file, generate the database, single protein running through your mass spec, and you are finally running it in your database through that particular uh, in a single patient. So that you can do. Or if you had a bunch of patients and if you want to study some kind of a population based study, then you can do for a multiple samples. Also you can find out that this person's having particular mutations is quite lethal, or they are not able to survive more, or some certain persons are surviving more. So what is the case, or some certain kind of global studies. So then you can do the one or multiple samples. So this is the G range objects and all those nitty gritties of the software, which helps you to the given. So VCF file, so that's what I was mentioning to you. Though the title mentions for the RNA-seq data specifically, and also the stress by the authors is on the RNA sequence data, but the VCF files derived from whole genome or exome sequencing can also be used to generate customized databases. That's what I was referring to. So don't no need to get very much anxious, or that if you have uh, exome sequence data or a genome sequencing data, or if you download certain exome sequencing data or the uh, genome sequencing data, then how you are going to analyze or how you can are going to generate the customized database, because the BCF files can be used to generate your customized databases. So now the junction analysis. So based on your input bit file that contains your splice junction. So from where the spice or the junction analysis came. So here, the as I was mentioning, that the authors stresses on the RNA sequencing data. So after you have done your RNA sequencing and you have generated you have done the sequencing and you have aligned it with your sequence, then if you try to find out if you need to find out, so you need to construct the transcriptome back. So then you can have the different aligners like your star aligners and all those, so based on your alignment tool. So some provides you the uh, option of having your so if you get certain so will help you to understand the novel fusions or the splice junctions or the certain things, so you can get from your analysis. So then you will be having those particular uh, data, so you can analyze those junctions into that. So three frame translated for all putative novel junctions. So if you're in the fusion, so what happens in the RNA seq, if you can understand that whenever a mutation happens, so sometimes there's a fusion between so which sequence or a novel exome sequence is generated as a result of that. So that exome sequence was where it should have been ended because of the fusion novel exome sequence. I am hopeful that you all are able to visualize and imagine that part. So a novel splice junction is created as a result of the fusion between two exon regions. uh, and then what happens is that you will definitely get a new sequence, or a new peptide sequence will be generated, which is a mutated peptide. So what we were looking for, or what we are trying to identify, a novel splice junctions or a novel mutated region, what we are further uh, uh, looking for. So we will be uh, uh, getting that. So obviously we will be getting that novel splice sites and the novel junctions. So now if you want to generate the database, so you will be doing the three frame translated peptide sequence for all novel junctions. So you can perform your three frame translation or the six frame translations. So if from the mRNA level, so you need to decide upon on the basis of your uh, understanding and what you want to achieve. We will perform the three frame or the six frame translation. So it's my understanding, or what I say, that I prefer to. So when I do from whole genome sequence or whole exome sequence to have a database, will perform the six frame translation uh, of the database. So how it looks that, so it needs a mRNA, so it will give a mRNA faster database. So once you get the faster database, you can easily put it up on your any sequencing search database for the proteomics datasets, and you will definitely get all those proteins or the peptides. So file which includes multiple samples, ANOVA annotation result files. So how will you annotate the files, the variance files? So that is being, so a popular tool is your ANOVA, which will help you to annotate your result file. So for making the database, you need to go on all those steps and need to understand and identify all those tools, which will give you the proper input format for custom ProtBJ to get it. So custom ProtBJ is not only the tool that is present, but uh, I prefer it, and mostly in the CP Tech papers also you will get this, because it is from the Broad Institute as well, and it is quite easy to use. So if you try it out on your own, you can get it, because it's actually beyond the scope for us here to do it, because all these you can understand that the sizes are very huge, and it takes a lot of time, and which is not possible to cover in a this short duration to show you the things. So that's why we are trying to give you as much as possible to close to that output, what input, what are the tools and the databases. So if you get any chance, by any means, by using your labs PC or access, or you download any data sets or come across it, you can try it on your own. So this is the prefix of the variant protein ID of the final output files: gene annotation data output reference protein database file, whether to add reference protein sequences to the output, and the verbose means it will, if you write this command of the verbose, so it will give you all the happenings, what is happening in your during your run of the tool. So so this is quite important for you to understand that what actually this tool or the software is capable of doing it and is not capable of doing it. So you don't over expect from it. So it can give you a customized database from based on single VCF file, based on multiple VCF files from a sample, based on multiple VCF files from multiple samples. So how now your question can a question can, or what I guess a question should come on your mind, that how multiple VCF files can be generated from a sample. If you have a single sample, then only uh, basic single VCF file should come, but multiple VCF files here means that from whole exome sequencing, RNA sequencing, both. So both will give you a multiple VCF files. So you have the looking at the genome level changes, at the translating transcriptomics level changes, and then finally at the protein level changes. So that this is how the format of the input variable find dot text you will be looking like the somatic germline sample RNA and multi anodes coming from the Hanover. So that's why if you do the Hanover, which is your Perl based tool, so it will give you the multi anode text. And then output file, what it looks is that varinfo.txt containing the detailed amino acid change information. So each row will be your variant. So each row will contain that this is variant. Var.fasta is your customized protein database, that what you are looking for. So what we are looking for is the protein. Yeah, this is also important, the summary data or the information, but we are can get it from the when we are doing annotating our variants using ANOVA, we can generate and we can actually identify all those variations. But the bat.fasta will help us to get the protein database or the protein sequences. So now let us so now what we have discussed is the in detail that how you can do the databases. So now you can understand there are the concepts remain quite more or less similar. So if you can understand the two approaches, one is for the custom prod PJ and what is being used across the worldwide for the cancer research for developing the sequences, and what approach we have used in for the uh, variant peptides for the COVID. So now, if you just think for a minute or for a moment, the differences between the two approaches. One major difference is that there was no alignment step which we did at the genomic level. If you all have noticed anyhow, uh, then you can find out that there was no alignment at the genomic level with the initial ones and then annotating those variants and then translating it. Rather, after translation, it was checked on with that which sequence or which frame is uh, correct one. Whereas here, there you need to have a alignment, you need to have a annotate the variants. So here what they are doing is that at only specific regions where the variations are there or the mutations are there, so they are creating that the variations can be introduced. So this software or this algorithm or this tool, what they are doing is that they are creating the FASTA file based on that variance at those regions where the variance can be present, so that the chances of your identifying the variance become increased. So so from now, so from these two examples, I hope you are can sorry, you would appreciate that the requirement is to have a database which will not provide any false positive data, false information, rather will provide us a very robust as well as the correct identification with less false identifications. So this is was the two major difference in the approach, but the custom prod BJ is being very much being heavily used for the cancer research, and you all can try to use that. So now, as I was telling, so there are some certain resources like DBSNP, Cosmic, Catalog of Somatic Mutations in Cancer. So I'm just giving you the name of the resources. So if you all can find out or can fill whenever you get time, definitely go and visit those websites or those resources, so that you can find out that how much there are like the 1,000 mutations database catalog of somatic mutations in cancer. Then there's a DBSNP database of single nucleotide polymorphisms. Then this is another one, which is your human cancer proteome variation database. So what it does, So till now, what we are doing is that we are trying to find out from our own samples, own data from the patients, clinical applications. We are trying to apply this aspect of the proteogenomics and trying to find out the exact changes in the sequences. So now, if we go on, one second. Yeah, so this is scan prover. What is doing that? It is designed to store and display single amino acid alteration. So one thing which I need to say, that what we are generally being very much focused and looking up to is the single amino acid alterations, because the single amino acid variations we are hypothesizing or uh, thinking that the single amino acid variations will cause the changes if it is in the it is not in the degeneracy of the codons. So again, the interesting fact is that the proteins not only it's changed to certain amino acids. If a single amino acid is changed to a certain similar amino acid with the similar physicochemical properties, then it is not so much deleterious or harmful. If a branch chain amino acid is replaced by a branch chain amino acid, it will have some effect, but it will not be so deleterious. I hope you all can would agree with that. But if a basic amino acid is changed to an acidic amino acid, or it is certainly a proline, or certain things come which is quite bulky, and all the structure gets protein structure gets distorted. So the scientists have uh, been trying to map the structure function relationship from a long time. So you can understand that the structure distortion would lead to the functional distortion. So that's concept. From that concept comes the single amino acid variations, or the single amino acid alterations, or the single nucleotide polymorphisms, which becomes quite important to understand for those particular diseases. So especially those related to the genesis or the development of human cancer, based on your published literature and the sources. So can prover, what it is does is that it takes on the what are the information, what are the resources, based on the literature, based on the sources, it takes on, and then try to map it and keep it so that you can easily. So we will see that how we can visualize those. So cancer-related variations and their cooperative can be guided protein names IDs in which user is interested. Furthermore, so if you all are not able to, if not you all are, but if anyone is not able to go through the steps of the genome sequencing data, and then finally mapping, annotating variants, and then trying to find out the first sequence, or then can their variation can be downloaded, as well as the searchable mutants for the shotgun proteomics. But you have the proteomics data, but you are still awaiting for your sequencing data. So what you will do with the proteomics data, or if you are not able to stuck in the analysis steps somewhere, so you can download for a preliminary point of view to check the coverage of your proteomics data. For example, you have done a, so these are all I'm trying to provide you the real case applications. So you have done a very fractionation of all those peptides, and trying to find out whether you are getting one mutant peptide or not, or whether you are at least getting something or not. So, and you are still awaiting your sequencing data, or you are in a stuck in a some step of the analysis, or analysis taking too much time. So, or you are downloading from the repository data, so anywhere you can be stuck. So you can here directly download the faster sequence of the proteins and their variation information, and you can use for the shotgun product. So if you are so for example, it can be an interesting thing. For example, you have induced mutation in a mice model, or in a cell line based model, and you are trying to find out that whether I am able to detect that or not. And you here, for that you don't need to find the sequence to find out that whether your mutation is actually working or not. So you can go here and can check for the proteins, their variation, and then directly you can add in the faster. But though that is not a quite a tedious task, because you can always go for that particular protein and you can mutate it on your own in a faster file and can search in your database. So but for those who doesn't know those things, you can always go for and try to find out. So this is the publication which shows the updated database for your human proteome variation. So this is again from the group of your Bing. So what it contains: cancer related variations, non-cancer specific variations, cancer related differential expression of proteins, gene and protein attributes. So it constitutes of all those things, and it is giving across the cancers, across the chromosome locations, across related proteins in pathways. So how what it contains: so for example, ensemble, swiss prot, gene names, refsec. So cancer type, protein list, chromosome location, cake pathway, cause cancer variations as I was mentioning, cosmic, OMIM, I guess you know, online Mendelian inheritance, these things, TCGA, Biomart, everything, DBSNP. So DBSNP is a non-synonymous. So I think we have discussed in detail about the synonymous mutation, non-synonymous mutation. So if you all just cannot recall, please go and back and have a watch at it, so you can find out the meanings of all those gene or protein databases. So in this can provide so cake pathways with CR, so the cancer related variants and the differential expression proteins highlighted, chromosome locations, the picture and mutation number statistics, gene name, mutation number in one protein of certain cancer type, cancer DPs, protein function, protein or gene. So everything we'll be finding. So what we will do is that let us go and look at it, that how it is there. Yeah.
Subject. The matches Baba.
Yeah.
Then the jargon.
So we can search here. So let us go by searching by cancer type. So let us take some head and neck cancer. So if we go for browsing, so you can see that all the gene names and the related variants, differential expression, everything is mentioned as it was mentioned in that paper. So you can easily uh, design. So you can see there is a point, so you can easily go and try to build up your hypothesis from here as well. So okay, so these proteins are showing differential expression, so these proteins are present in large number in any cancer samples. Then how you can look forward and take it forward from there, that also can be done. So here you can see that you have it will mention about your ID that you have entered, protein sequences, UNC homologs, chromosome 7, gene ontology that contains your biological process. So change in conservations, domain, cancer name, melanoma. So you can see data source you can get. So as I was mentioning that they had shown that it contains from your all your cosmic DBSNPs and everything. So then you have your
Better
uh,
options.
variation that in the 100th position is 100 has been changed to G. Yeah, I'm audible, right?
Yes, you are audible.
Okay, so then is your Q154K and all those. So here they were mentioning about the proteins that you can download the validated DBSNPs, cancer resistant NSNP. So you can have your uh, from here the cancer related head and neck, lung cancer, pancreatic, renal. So anything you can MS can provide data. So you can easily go and download it. Also, you can search for the protein list for some pathways mentioning. So you can go and submit any pathways as well. So you can find out with cancer related differential expression and both. So you can easily go and find out. So you can explore it as well as you can explore the other mother databases from where the data has been taken. Okay, so that will again uh, provide you much more information. So now what we were mentioning that the VCF file is quite important. So because it will give us understanding. Firstly, is that how much our files are
Why?
aligned, annotated. Yeah, Amit, are you saying something? Yeah. So first thing is that how the VCF file, firstly from first point of view is that annotating the variance and identifying and understanding our variance. And the second thing from point of view is that updating the database. So what is the file format actually looks like, or how will you understand that the VCF file that you have generated is correct or not? So this is an example of a VCF file which we will discuss. We will look into different aspects of it in a little bit detailed manner. So you can see that the first is the file format. So this is the mandatory header line. So you can understand the FASTA format doesn't will FASTA format will always contain uh, greater than sign, without which it cannot be considered a first file. Similarly, for the VCF files, it should be having a mandatory header line, because whenever you feed for any input, so if the software or the algorithm will firstly look for the header line, and when the format becomes okay, it will go on to the next thing. So this is your header. So these are all optional with numbers and all those things. But then the body, which is your important one, is your chromosome number, position, ID, reference, and the alternate. What is your reference is ACG, what is your alternate, either AT, insertion. Okay, qual filter, whether it is passed or not, info. Then is your samples and the reference alleles, alternate alleles. So deletion, SNPs, large structural variations, insertions, other events, all these things will be there. So this is a general format of how VCF files look. So here, so now the question comes that when your VCF file is generated after you annotate your variants. So what is the annotating? This is the annotation. So what annotation? What we are generally calling upon is that whenever you are aligning your sequences. So this is your reference sequences, and so what you are doing is that you are annotating this variance. So the tools or the software that use for annotating the variance like the ANOVA just performs this kind of annotation. So in a VCF file. So this is a very good article, I would say, if you are much keen and interested to learn really about that, understand that how those VCF file looks upon. So you can look at how the small insertions, large deletions, and all those different structural variations are incorporated into the sequence. You will get benefited from it. So the variant call format VCF version 4.2 specifications. So so you can go and find out in this link the VCF format specifications. So which you will be getting aware of it. So now let us look at some important aspects of from this article which we will be discussing, for which will be important to understand. So the VCF specification is that the VCF first of all is a text file format. So whenever we are trying to dealing with this kind of big data analysis, so we need to understand that what is our input file, output file, and the file formats that we are using, without which it becomes impossible for us to work. So it is a firstly it is a text file form. So you would not be able to understand that where is the error or how you are getting that result or file, if not you have used a you know the what are the actual file formats are required and how you are going to be using that. So it is your text file format. It contains your meta information lines, a header line, and then the data lines, each containing information about a position in the genome. So if any of these things are missing, you would not be able to process it further, and you would think that why my data is not getting assessed. Okay, so this is quite important for you to understand, and also has the ability to contain genotype information on the samples for your each position. So you have, for example, file format. So as we were mentioning, so the chromosome. So now I think you can understand, right? Position, ID, reference. So now we will be looking into some important aspects. So that example which we show, which just showed, is a good simple SNP, a possible SNP that has been filtered out because its quality below is 10. Okay, so if you have see that the filter Q10, here you can see. So here this also needs to be mentioned. So if you use a filter of the Q10, so it will get filtered it out. Has been filtered out its quality is low, at which two alternate alleles are called, which of them T being ancestral, possibly a reference sequencing error, a site that is called monomorphic reference, that is with no alternate alleles, and a microsatellite with two alternative alleles. Okay, so these terms and to understand all these things are important. One a deletion of two bases and the other insertion of one bases. T genotype data are given for your three samples, two of which are phased and the third is unphased, with per sample genotype quality, depth, and haplotype qualities given, as well as the microsatellite calls are unphased. So the which are the microsatellite calls are generally called your unphased. So that's why you need to go through it. So I would recommend you to go through it the detail. Then you will be able to understand much more. So from that VCF file only you would be able to understand that what is the uh, variance and all, and you will not rely on any tool or software, or you will not be able to, it's not happen that you won't be able to figure it out what's the cause. So file meta information is included, key equal to value pairs. That information info filter format in the VCF file be included in the meta information section. So this is the talking about this particular sections. So a single file format field is always required. So here it should read this. So this is very very important. So info field should be described as follows, which you will know that it should be have the number, type, description, source, etc., etc. So the possible types for info: integer, float, flag, character, string. The number entry is an integer. So you know all these things. So filter ID description. So as you can see that the filter quality, because of that you can remove that if you want to remove certain IDs or certain chromosome numbers or the positions that you know that it is there or something like that, you can do that. So alternative allele field format. So all these format should be maintained. So if you are using some other tools and not using ANOVAR and still can make this kind of field format, then also you can able to go ahead with your database generation. So inversion, copy number, loop tandem, you all need to you can go through this individually. So now the header line syntax. So this is very important. So this is important in the fact that the header line names, the 8 fixed mandatory columns should be there. So it column should be the hashtag chromosome, position, ID, reference, alt, quality, filter, info. So without these 8 mandatory columns, you cannot move forward. So this is quite important. Without this, we cannot move forward. So if the genotype data is present in the file, column header and arbitrary number. So duplicate sample IDs are not allowed. So that's why whenever you are taking for the multiple sample VCF files, so it should contain different IDs. So additional information, you can have ancestral allele, allele counting genotypes, AF, all these things, membership in 1,000 genomes, validated or not, validated by follow-up experiment, somatic mutation, etc. etc. You can do that, but the major thing is that the 8 lines should have the uh, mandatory columns should be there, and info keys used for your structural variants and all those things. So now this is again important part, is that the creating your VCF entries for SNPs and small indels. So how you will be recording for your small indels, insertion deletions, or the small SNPs. So SNP polymorphism, you can give it that CG. So of C, changing of C to G is given by this second bracket. You will give the CG. C is the reference allele and G is the alternative allele or the changed one or the mutated one. A single base deletion of the C is will be given by TCT. So TC is the reference allele. Insertion of A is TC, TCA. TC is your reference allele. So position, reference, alternative. So you can see that the TC, pass, filter, and the info. So this is how you need to incorporate your small indels or the small changes into a VCF file. So this is so if you all try to, if you are understanding and if you find out some shortcomings in the ANOVAR or any those mm, tools which are, if you feel that it is overlooking certain aspects of the biological aspects, which, and you need to design a software tool, you can do so, but you need to keep in mind the annotations, how that needs to be, the software or the tool needs to incorporate in the VCF file, which can be taken forward for further analysis, otherwise it was not possible to do so. So this you need to specify your complex rearrangements with the breakends as a set of novel adjacency ties together to your two breakends. Uh, so if you breakends happen of a novel adjacency, then it's called the mates. So this so the, if you see that the, that is one record for each of the two breakers, when you will find out that it is a novel adjacency or not, so there is one line of VCF, that is your one record for each of the two breakers, then you will find out that it is okay. So the tag will also mention that is a breakend here. So these are called the novels. Then the string T that replaces places of your S, and so this is the meaning. So P is extending to the right of P is joined after T. So reverse from P is extending left of P is this uh, this software, these brackets and those things becomes quite important to understand. Left of P is joined before T, so this you can understand. So this is how you will do see break-ins. So if it is joined from uh, here to here and all possible orientations of the break-ins. So in the in that link what I had shown just now, you can go and have the detailed understanding of all the parameters of the VCF files, what is there, and then you can definitely try to understand or make meaningful out of it. Uh, so then you can definitely go and find out the what is the actual meaning of the VCF files and can read VCF files on your own. So now moving on. So as I was mentioned that now we had, as we mentioned that we have completed firstly the integration of the how we can integrate the parts of it, genomics, proteogenomics, and how the database can be done, housing keeping a VCF file. So now what we are looking at the genomics viewer. So that what I mentioned that how we'll be looking at the visualizing the genomics uh, in the sequences, the chromosome numbers, the sequences after getting, if you can on your own that the insertions, deletions, etc., etc. See if you have a bed file or if you have an SNP file, you can uh, upload that and can visualize it. But this is a very good software which you can download and use as well as in the web version. We can use that as well. So integrative genomics viewer help you to analyze your lookout for certain things. So this is the user guide. So you can go and download integrative genomics viewer as well. So after you have gone the variance file and all those, but you cannot you are just in a text file format, you cannot read that, or it's not possible. So you can visualize, always prefer to visualize in certain aspects. So IGV helps you to for a very good visualization and a better visualization of it. So, so let us go for the online one and try to see that whether we can use it or not. So here, so firstly this is the interface. So you can upload. So they provide you the option to upload from Google Drive, Dropbox, or anything. Also the different human, other organisms, etc., etc., present. Okay. So you can go for, so as we are mentioning, AG38, GRCH 38, AG38, which is the most probably the latest one, or using AG19 or AG18 or mouse or sequence database. So let us go for the AG38. So it's changing here. You can see. So you can see here that it got changed. So this is now all. So you can see that 1 to Y, chromosome 1 to Y, everything is there. But it's not making sense. So let us try to look at for certain things. So if we go for chromosome 3, so what is happening? So now there is containing all the databases, although base pairs. So now if we go and try to find out 11000 to So okay, so I'll just remove one. So yeah, so what we are finding is that, so now we are looking at a certain stretch of the 46 base pairs. So now these are the refsec genes. Okay, so now you can understand this is the portion where the genes are there, means it will produce your functional protein or functional RNA at that level. So the gene, this is a 46 base pair length particular stretch what we are looking. This is the particular gene part which is, so here all are your triplet codons. So now you can try to see by that by moving on by if you incorporate certain uh, changes to it, then how the protein level will get changes and all these things, so you can find out. So here you can see that you can go here, you can go come here, so you can stretch it out and all the sequence you can do it for the HG38 thing. So see, it is going. So again thing is that, so you can save it and all those things. So yeah, so this is in the circular view also. If you want to visualize your genomic sequences, you can do so. So you can change it, you can go to any of the chromosomes you like. Say if you want to find out any particular things, particular mutation, you can easily go and do it. So it's a very uh, easy to use software, but you can very useful specifically to check out your variants and to identify your variants when you are using this tool. So you can definitely go and find out the variants. Also you can find out, the simple experiment you can do is that to find out the variations between your HG38 and the HG19 for a particular chromosome. What is the difference? So HG38 versus HG18 versus HG19. So that will also, for a particular chromosome, just you take randomly and try to find out whether there is any difference or not. With also you can go ahead and read about HG18, HG38, and what is the major difference, which if you look at it out and then try to find out whether you are able to visualize in IGV or not. So this will give you a feeling of that you can visualize the sequence. So whatever we are talking about, the text of the sequences or the strings of the sequences, whether you are finally able to you will be able to the visualize in the form that we generally look in the different textbooks like the AGT, ATG sequences and all. So you can find it out. So now, so now finally, so as I mentioned that after going through it, so we need to have a understanding of that what are the proteomics technologies that are being used and how the labeling technologies helps you to identify your further so increasing the coverage is one challenge, because the genomic sequences give us all those peptides and the protein information. So again, I think you all would appreciate as of now that the fact that the genomics, though the genomics technologies gives us, the proteomics technology still needs certain uh, improvement in the terms of coverage, accurately quantifying to reduce the false discoveries. So that's why it's important. So the accurate quantification technique one is your, so I'll just recall or brush up those things once more. Is that the firstly is that if we look about the proteomics technologies and the quantification strategies, the first important strategy is the level free quantification, where no labeling techniques are used and the spectral counts or the intensities or your uh, the spectral match or the peak to peak match is matched, and protein is accurately quantified from the peptides. But another technologies or that are being used is the labeling technologies, where you try to label your amino acid. So what you will be labeling, either you can label the whole amino acid, a single amino acid, or you can label a particular peptide. So upon depending upon that, so you are there are different labeling technologies for the accurate quantification of your particular proteins. So I track is your isobaric track for relative absolute quantification, which is your in vitro labeling technologies, along with your uh, TMT. So these, so if you go by the name, you can see that isobaric. So because your M by Z's are uh, uh, that matched in the mass spectrometer. So if the same amount of masses are added to each condition, to each peptide, then there will be no effect. So that's why sample A, sample B, sample C, sample D, that is your different conditions. The peptides are labeled with 114, 115, 116, 117, but the balancer and the reporter, so the balancer group will uh, just adjust your weight balance and will make the isobar, that is the same masses. And then the peptides are labeled, so then you can see that the reporter ions will be quantitative. So 114, 115, 116, and 117, and on the basis of the peaks or the intensity levels, so you can identify that what are the changes in the conditions, or accurately quantify, relatively quantify that which one of your protein is highly quantified or highly expressed in your particular condition or not. Similarly is the is another labeling techniques is the ICAT, which contains your, which tries to label a particular amino acid. And then so it will not label your peptide based labeling, but your amino acid labeling. So sample A will have the light labeled, and in the heavy isotope level sample B will be labeling. You will be mixing, and then you will be performing the analysis and try to find out that the heavy labeled and the light labeled, what is the difference in the quantification level. Similarly is for the so another option is the Silac. So Silac is, what is major difference between Silac and iTrack and your TMT is that the Silac is uses, it is an in vivo labeling system. So if you are asked to have a culture which you need to perform in the cell culture based experiments, means if you are using for example, you are culturing a cell and you are giving a drug treatment, or you are knocking down certain genes and trying out to find out what's the difference in the expression level between these two conditions of the cells, and want to quantify that, then you can use an in vivo labeling system, that is a Silac. So what you will do is that for one condition of the cell, you will have you will grow in a you will means take it out an essential component of the amino acid from the media and replace it with a heavy labeled amino acid. So in one condition will grow in a normal media with light labeled, in another it will be growing with the heavy amino acid. So after 3, 4, 5, 6, 7, so after a number of passages, what will happen is that the heavy labeled amino acid will get incorporated in your cells. So for a particular condition which grows in that heavy amino acid, and if you treat that cell with a drug, so all the amino acids will be in a heavy labeled, and for your other states will be in your light labeled. So what will happen is whenever you extract your protein and perform the analysis, then you will get that for that particular condition all the heavy labeled amino acids. And by matching your heavy labeled amino acid by the light labeled amino acid, you would be able to detect or relatively quantify what is the differences. So this is for the stable isotope level for the in Silac, or the immuno or the in vivo based labeling methods. So this just for refreshing the different quantification strategies that are present. So once you get your peptides, you would try to quantify that in your diseased case, and in what your case, what is the difference. And you can use your appropriate strategies on basis of your, if you want to do a in vivo labeling or a in vitro leveling. And of course you can also do an in gel gel based proteomics, in the sense not gel based, but you can run on your gel and you can find out certain protein bands which are of your interest, which you need to quantify, and then you can perform in gel based digestion, and then you can perform in the mass spectrometry. So this is a general overview of you can perform either in vitro leveling with multiplexing. So if you just ask, or if your question comes that what is your major difference between your level free quantification and the level based quantification, is that so for the level free quantification you need to run your samples every time with increased chances of inducing variations, which you need to tackle it later. Whereas if you pool the level based techniques, you can pool your samples in iTrack or TMT, and then you can move way forward, and with a single run you can perform the analysis of the or the experiments. But the cost of the labelings will always remain associated with it, and it will become much more cost uh, uh, costly rather than the level free quantification. But it will always depend upon what experiments you are trying to follow. So after which you will perform the MS analysis and the data interpretation. So just summarizing of what we have discussed in today. So if we just go ahead. So this is important thing where we need to understand that how, why. So we have discussed in detail that why customized database construction is the key role in all this process of integrating genomics and proteomics data. So individual technologies having giving much better results and reporting much greater things, but overcome of the limitations could be done with the help of this customized database construction. Then we have discussed that how the databases can be done, and with a case study of how we could help in detecting certain mutant peptides in COVID with the help of this simple concept, which can be so extrapolated from the cancer to the any infectious diseases, and for any kind of those microbial and epidemic diseases. Second is the custom product, which is for the RNA-seq, but you can do from whole genome and whole exome sequence also. With the VCF files, you can generate the customized databases, and that's why we discussed in great amount the VCF file format, because it helps us in annotating the variants as well as in the database generation. Then followed by what we did is the some resources like the Cantrovar. Also, I recommend to all to go for the DBSNPs, Cosmics, catalog of somatic mutations in cancer to look for your particular protein of interest or gene of interest, to look for that what are the variants databases are available. So this is one of those Cantrover, and also we have shown that you can easily download the data faster database if you are not willing to follow the steps and generate the customized database, or you don't need to do that. You can always go and easily download and try to search in through the database and find out that what is the FDL value, Q value. If you want to experiment, then you can do that and try to find out what are the results. That if you are analyzing sequence data downloaded from SRA, sequence read archive, and you are generating the database, and whether when you are giving to the Uniprot database, so what are the different results. So you can definitely go and find out. Then the PCF file format, what is the headers, what is needed. And finally we have looked upon the IGV, integrated genome viewer, to visualize the things. And then finally with some labeling techniques, some brushing up for the proteomics labeling technique. So do you have any question? Anyone has any question? Or